Hello, my name is Juliane Damm. I'm a PhD candidate at the Instituto de Neuroetologia de la Universidad Veracruzana in Mexico. Together with Colleen Gault, I went out on a quest to find a non-invasive measure of acute stress response in wild female white-faced capuchin monkeys, and I would like to take you on that little quest with me. The 30 years of research on wild capuchin monkey social behavior at Lomas Barbudal have revealed that white-faced capuchin monkeys show an intriguing complexity of social behavior. This kind of complex social behavior elicits the interest of behavioral endocrinologists who try to understand the relation between hormones and behavior. One branch of behavioral endocrinology specializes at looking at how social behavior modulates the so-called stress physiology of an individual. When working in a wild setting, non-invasive methods for measuring stress are preferable because they minimize the interference with the animal's natural behavior. Glucocorticoid metabolites as indicators of the stress physiology can be extracted from urine and feces. Capuchin monkeys are relatively small-bodied, mostly arboreal monkeys. The collection of urine is thus difficult, and the collection of feces becomes the method of choice to investigate the stress physiology. As feces in most species integrate glucocorticoid levels over many hours up to several days, they're usually considered an integrative measure of stress. That is, they are used in studies that look at prolonged periods of challenges and stress events. However, capuchin monkeys are different. Capuchin monkeys are not only stars when it comes to social behavior. They have a short gut transit time combined with frequent defecation up to 24 times per day. There's also strong evidence that glucocorticoids in response to a stressful event are excreted fairly quickly in the fecal matter. In 2 studies, the peaks of glucocorticoids in response to an ACTH challenge and a vet examination could be tracked in feces as soon as 2.5 to 4.5 hours after the onset of the event. In our study, we thus set out to do something very different and very bold. We asked ourselves whether glucocorticoids measured in feces could be used as a time-matched measure of acute responses to stress events in white-faced capuchin monkeys. In order to do so, we investigated whether we could pinpoint a response to severe within-group aggression and intergroup encounters in the continuous record of fecal glucocorticoid levels over an entire day. We assume that severe within-group aggression and intergroup encounters are events that are likely to elicit a glucocorticoid response. The data presented here was collected on 11 female white-faced capuchin monkeys at the Lomas Barbudal Reserve close to Bacasses in Costa Rica. Behavioral data on incidences of aggressive behavior and intergroup encounters were collected as part of day-long continuous focal animal samples. The data was collected for at least 2 consecutive days. Throughout the following day, all possible feces of the focal individual were collected. The feces were frozen until they were oven-dried at the field site. The samples were then shipped to the Max Planck Institute of Evolutionary Anthropology in Germany, extracted according to the Weltring et al. Protocol, and then quantified using a cortisol-specific enzyme immunoassay as developed by Munro at UC Davis. In order to aid your understanding of the results, I will briefly explain a hypothetical example of 1 day of data and how we associated fecal hormone measures with behavioral data. Here, we have a line representing a continuous focal behavior data set for 1 day. We further collected all the feces that the individual deposited throughout the day. We then identified events of severe aggression and intergroup encounters according to predefined criteria within the behavioral data. And we also generated a control event unrelated to behavioral data, and therefore expected those control events not to have any relation to the stress levels. According to the results from the previous studies, we hypothesized that the longer the lag time between the onset of the event and the time point of defecation, here T0 would be the onset of the event, and T1 would be defecation of the first sample, and T2 would be defecation of the second sample. So that T2 is basically the lag time, and T1 would be the lag time of the first sample. And we hypothesized that the longer the lag time between the onset of the event and the time point of defecation, the higher the glucocorticoid levels, until a peak is reached in the 2 to 4.5 hours window, followed by a decrease with yet longer lag times, such that fecal glucocorticoid levels are basically a function of lag time. To account for the expected rise and fall of the fecal glucocorticoid levels in function of lag time, the variable of lag time was squared. With the data as described, we built a generalized linear mixed model, including fecal glucocorticoid levels as the dependent variable, the interaction between the event type and the lag time between the onset of the event and the time of defecation were entered as a predictor variable. We further controlled for season, time of day, and infant age. Running this model, we obtained the following results. There was no significant effect of the interaction of event type and lag time squared. There was also no significant effect of season. We did, however, see a significant effect of time of day, with fecal glucocorticoid levels being higher in the earlier samples, and we saw a significant effect of infant age, with fecal glucocorticoid levels of the mother increasing with her infant's age. Since we did not see a significant effect in our predictor variable, we computed the 95% confidence intervals using 1000 bootstrap replications of the mean fecal glucocorticoid levels for each event type. The graphs show the log transform fecal glucocorticoid levels on the y-axis over time after the onset of an event, x-axis, and that for each event type. The dotted lines indicate the upper and lower 95% confidence interval. And as you can see, the mean fecal glucocorticoid levels stayed within the 95% confidence interval for the control events in the two types of severe aggressions. However, the mean fecal glucocorticoid levels 2.5 to 3.5 hours after the onset of an intergroup encounter did fall outside of the 95% confidence intervals as we predicted. Analyzing the results, we first wanted to stress the limitations of the study, one of which surely is the sample size. With an increased sample size, especially when looking at the intergroup encounters, we might have been able to reach significance levels. However, the data set is just a subset of a bigger data set and was supposed to be a pilot study. That's why we opted for a smaller sample size. We also questioned whether there was maybe a flaw in our hormonal quantification. However, we ran several quality controls that were all within the generally accepted margins, and we further were able to pick up a well-known biological effect, the daytime effect. And for many species, it has been shown that glucocorticoid levels increase just before awakening in the morning and decrease throughout the day. Since we were able to pick up this effect, we think it's unlikely that our hormone measures as such were flawed. There could also be a biological explanation for the obtained results. One possibility is that capuchin monkeys express an insufficiently large physiological response for the selected stressors that can basically not be tracked in the feces. Another possibility could be that the frequency of behaviors that could possibly elicit or mitigate a glucocorticoid response are so frequent in capuchin monkeys that even frequent defecation is not enough to trace the changes in fecal glucocorticoid levels. So in conclusion, when looking back at the original research question, we can say that our methodology was not able to pinpoint responses to the events of severe aggression or intergroup encounters with statistical significance. Therefore, also the more general question of whether fecal glucocorticoids can be used as a measure of responses to acute events of stress remain inconclusive. With this conclusion, I come to the acknowledgements. I would like to acknowledge, or I would like to thank the academic supervisors, Susan Perry, Tobias Dechner, Roger Mundry, and Franka Cebus. I would also like to thank our lab technician, Vera Schmeling at the Max Planck Institute, and Don Cohen for his marvelous work with the databases. Colleen Gault collected all the physiological data and behavioral data for this study, and she had some marvelous field assistants that helped her throughout the data collection. So a big thank you to Garrett, Christine, Jan, Keith, Beth, and Jason. Funding was provided by the National Science Foundation, National Geographic Society, Leakey Foundation, and Graduate Women in Science. These grants were awarded either to Susan Perry or Colleen Gault. With this, I would like to thank you for watching the presentation. I hope for an interesting discussion.